Um, so welcome everyone. Thanks for joining us for this month's ACEAS seminar. Um, my name is Kieran Murphy. I'm the UTAS sort of seminar representative, and we also have Yuha at ANU and Fabio at UNSW. So just a reminder that if anyone has any visiting speakers or if you have any uh, work you'd like to present, please do send us an email. We've got a few talks lined up, but there's spots later in the year. And so before we get started, I'd just like to, on behalf of the University of Tasmania and also ACEAS, acknowledge all of the countries that are participating in this online meeting today, and to acknowledge their elders and ancestors and the legacy that they leave to us. Okay, so I'm just going to pass over to Professor Strutton now to give a bit more of a personal introduction to Tamara. So thanks, Pete.
Yeah, thanks, Kieran. Yeah, so the speaker today is Tamara Schlosser. So Tamara is a research associate in ACEAS at the University of Tasmania. She has degrees in environmental engineering, a bachelor's and a PhD from the University of Western Australia. And in between those degrees, she worked in industry for a while. And then after she finished her PhD, she was a postdoc at Scripps Institution of Oceanography in California from 2019. Excuse me, until 2022. And then she started with started with ACEAS, where she's been since. So she's actually recently moved back to Perth, where she comes to us today. Sorry about that.
Thanks, Pete. Okay, so I'm probably going to talk today about different physical controls on the Southern Ocean spring bloom, which I've worked most closely with Pete Stratton. But the first segment of work I'll discuss on there we go, is work I did also closely with Kerry Lee Baker and Philip Boyd. And in this work, I was really looking at how light and mixing controls the spring bloom onset. And so if you're not familiar, the onset of the spring bloom is when biomass first starts to increase. And so it tends to also be when biomass is at its minimum, and then biomass increases, reaches its maximum biomass around spring or summer. And so we're interested in what is controlling the very start of this increase in biomass, particularly because if there's a change in the physical environment with climate change and the timing of these seasonal events changes, it can have big impacts on higher trophic orders, for example, things like krill dynamics. But there's two main theories in the literature of what controls the very start of the spring bloom. One of them being the bottom-up theory, which is where it's the availability of light and nutrients, which is constraining the growth of phytoplankton and the accumulation of biomass. And so bottom here is bottom of the food chain, not depth. And so this is consistent with the Sverdrup's critical depth hypothesis shown here, where it's theorized that it's the shoaling of the mixed layer around the springtime that results in an increase in light availability and therefore an increase in the growth rate of phytoplankton, and the biomass accelerates in concentration and the bloom occurs. And so this contradicts with the top-down process that is theorized by a number of Biomass papers. This one's called the disturbance recovery hypothesis. And in this theory, it's the deepening of the mixed layer due to an increase in the mixing rate that is predicted to decrease predator-prey interaction. So essentially, it's harder for zooplankton to come into contact with phytoplankton. And because of that, they predict that the grazing rate decreases. So the mortality of phytoplankton is changing. And because of this, biomass is able to increase. So two very different schools of thought as to what is driving the start of the bloom, and therefore how phytoplankton are going to respond to changes in the physical environment. And so both of these theories assume that there is a well-mixed mixed layer, and the details of active mixing can modify these theories as well, which I'll get into later. So I'm focusing on just the Pacific sector of the Southern Ocean, where we have over 70 BGC Argo floats, and we are able to use that data to identify over 180 spring bloom onsets distributed across the map. So I mainly look at things like the temperature salinity from the CTD, as well as the biogeochemical observations, such as chlorophyll fluorescence, to identify where photosynthesis is possible, as well as looking at the optical backscatter, which I can use to estimate the phytoplankton biomass. So it's kind of like a carbon-based estimate of biomass that I'm able to use. And so the details of the mixing matter in the Southern Ocean, because on average storms occur around weekly. And if you look at observations of both density and chlorophyll fluorescence, this paper by Magdalena Carranza highlighted that most of the time, the depths where density is well mixed is not the same as the depths where fluorescence is well mixed. And so what this means is that the depth of active mixing, where everything is well mixed, all the phytoplankton are evenly distributed, it can be different to where the density is well mixed. And so I differentiate these two ideas by calling the region where the density is well mixed using traditional definitions as my mix layer. And then I define my mixing layer as the depth where active mixing is occurring, and phytoplankton are well mixed throughout that layer. And so I want to know things like how quickly are they well mixed, what is driving that mixing. And so to understand that, I use a 1D model called the General Ocean Turbulence Model. So I set this up with BGC Argo stratification, and then I force it with Error 5 atmospheric reanalysis data. So then we have an idea of what is controlling the variability of phytoplankton. So in the simulation, it's able to accommodate a large number of different processes that control mixing. For example, heat and moisture fluxes, where if the ocean is getting cooled in the wintertime due to negative heat fluxes, that causes convective turbulence, as an example. However, I do use a simplified approach here. So I don't consider a effect of surface waves. So I don't include Langmuir turbulence in my simulations. And I also don't include 3D effects. So things like fronts and eddies can either suppress or enhance turbulence, but I don't consider that. But despite the fact that I'm using a more simplified approach to understand mixing, we still do a really good job of estimating the change in mixing depth, where we can make an estimate of where mixing is occurring in observations and compare it to the model. And so we do a really good job of predicting that, but we just tend to overestimate mixing depth by about 20 meters on average. Okay, moving on. So here is a map of when onsets occurred. So it's colored by the month. So blue's in May or June, and then red's as late as October. And so you can immediately see there's this big latitudinal trend. And so what I've done is I split it up based on the ACC fronts. And so the southern barrier for the northern region is the subantarctic front, and then the northern limit for the southern region is my southern ACC front, that thick grey line, and then middle in between. And so there's this really evident latitudinal trend with earlier onsets to the north, but there's also this longitudinal trend with earlier onsets to the northwest. So something else is going on there. We also noticed that there were much later onsets under sea ice, where all the points with a black border had sea ice present when the onset occurred. And so immediately you can see a few key environmental drivers that might be occurring. For example, the availability of light and how that varies in the latitudinal direction. But we looked at a large range of different variables that might be controlling the bloom onset. And so the way we looked at what might be influencing all 70 odd floats is by using the date of the onset as our consistent, like x equals 0 here on these figures. And then to the left, I'm looking at up to 2 months before the onset, and then to the right, 2 months after the onset. And so the idea being that if we average across all of our floats, there should be some common trends in how the environmental drivers are changing, and then these will relate back to the bottom-up or top-down theories behind onset drivers. So here at the top, I'm showing the mixing layer depth. In the middle panel is the average light availability over the mixing layer. And then in the bottom panel is our mixing rate from the model. Okay, and so I'm showing just the northern data here. And immediately we can see that there's this deepening trend of the mixing layer, where it's at its deepest 5 weeks after the onset. Light availability continues to decrease around the time of onset because of that deepening of the mixing layer. And then the maximum mixing rate is observed a month after the onset. So this is not consistent with the bottom-up theory, which states that light availability might be controlling the start of the onset. Instead, it's quite consistent with this top-down control or the disturbance recovery theory. And so in this, it's that increase in mixing rate and the reduction in the grazing rate, which is driving accumulation of biomass. However, I'll note that we have no direct observations of iron, and so we don't know the depth of the ferrocline or where nitrate becomes available in the water column. So we can't fully discount the influence of iron in our observations. Next, if I add on the lines for the middle and southern region, you'll see that there's a trend in the south, at least, this blue line, where the maximum depth of the mixing layer is at the time of onset, and it's a bit more inconsistent in the middle. But then when you look at light availability, because of changes in the surface light, light is consistently increasing around the time of onset by multiple orders of magnitude in the south, but it's still definitely increasing from a month before the onset in the middle region, steadily increasing. So this is definitely consistent with the bottom-up control, where an increase in the light availability is influencing the start of the bloom. And when you look at changes in the mixing rate, they're not as consistent as compared to the northern region. So it's definitely less likely that the disturbance recovery or top-down controls are important near the poles. We also looked at the variability of other variables, and one where we did find significant correlations was in the trend in mixing layer temperature. So this left plot shows the mixing layer temperature at onset with the onset date on the x-axis. And so it's coloured by the latitude. And so there's this trend of earlier onsets in waters, and we do know that temperature has an impact on the physiology of phytoplankton. So for example, they're able to grow light harvesting pigments, which lets them more effectively turn light into energy. So they're able to do it better when it's warmer. So we know that there is some experimental data showing that this trend is real. However, if you look at the trend in latitude, it's very obvious that latitude is also changing in this plot. And so there's correlation, not necessarily causation in this left plot. However, if we look at the right plot, this time I'm plotting temperature by the longitude. And so here in the northern region, we can see that the reason why those northwest points had an earlier onsets than the northeast points was probably because of temperature. So in summary, yes, things like latitude, day length also impact the onset, but we can't fully discount the role of temperature in our results. And if temperature is controlling the start of the bloom, then we can make some predictions of how onsets might change in the future. So in the more northern regions, there's a prediction or observational evidence of waters getting warmer every decade. And so this would encourage a shift to earlier onsets. And then in contrast, in the south, there's a small trend of waters getting colder. And so this would encourage later onsets in this region. So to summarize, I've used a novel combination of BGC Argo and a 1D model to try to determine the environmental drivers of the Southern Ocean bloom onset. We found that the mixed layer is a poor approximation of active mixing in the Southern Ocean. And so instead we focused on the variability in this active mixing layer in our analysis. In the northern region, we found that dilution of the mixing layer and decreasing predator-prey interaction likely led to a decrease in loss rates and grazing of phytoplankton, and that allowed biomass to accumulate and the bloom onset to occur. But in contrast, in the middle and south, we saw this quite dominant control of light on net growth and on the bloom onset. And this is obvious in this panel here, this figure, sorry, where on the y-axis is the accumulation rate, where the first time the accumulation rate is positive is when the onset occurs. And then on the x-axis is the light availability. And so there's this strong linear correlation indicating that as soon as light hits some certain threshold, the onset is able to occur. In our manuscript that we've submitted and still waiting on feedback, we also look at the role of phytoplankton physiology, temperature, ACEAS, as well as iron availability, which I only briefly touched on here, and how these all constrain net growth in all regions. So this links on to some work I've been doing with Kirili Baker and Philip Boyd, where for Kirili's postdoc, she's been looking at the impact of phytoplankton physiology and intermittent mixing on and how that impacts growth. So in essence, I'm using my BGC Argo observations to help inform experimental data, and through having a better understanding of how phytoplankton are impacted by light and mixing, we can then use that to better inform our field observations as well. But I won't go into that too much today. I'll just take a drink of water. Okay. So in this next section, this is what I'm currently working on. So it is a work in progress and is a little less polished. But Pete and I have been looking at how phytoplankton blooms are impacted by high or low sea ice extent. And this work is really motivated by recent publications like this one by Ariane Purick and Ed Doddridge that was studying the critically low sea ice extents that occurred in 2023 and how different physical and biological processes might be impacted by these low sea ice extent. And so from reading similar sort of papers, I had these ideas like how is the 2023 phytoplankton variability impacted by this recent low sea ice extent year? And is there other trends of phytoplankton variability being impacted by low or high sea ice extent in our observational records? Can we observe higher trophic order impacts? And are we able, with our current sampling, to observe impacts that are occurring? Or perhaps we need to modify our monitoring schemes to understand how the Southern Ocean is changing. So to start with, I needed to define my marginal ice zone. And so to do this, I found the maximum extent of ice every year since 2014 from the ERA 5 data, and then defined my AvMax ice extent as the average of that maximum extent since 2014. And so that's really just this outer black line here on the plot. And then here I'm showing the distribution of core Argo and BGC Argo floats for different years, where core Argo has just a CTD. It lacks any of the biogeochemical sensors like chlorophyll fluorescence, for example. And you can look at the relative number of these floats on this plot, where the number of profiles on a log scale is on the y-axis. And so you can see that the number of total floats has been relatively steady at increasing to around 200 floats in recent years. But if you look at just biogeochemical floats, we have a lot less. So since 2015, around then, we've had around 40 floats. But before then, we have very few, unfortunately. And I say unfortunately because if you look at 2015 and later, that's when the sea ice extent anomalies become more extreme. And so unfortunately, we don't have any BGC data from at least BGC Argo profiles before 2015 around. But from the sea ice extent anomalies shown here in the circumpolar in black and in specific regions in the bottom panel in color, we define certain years that had particularly high sea ice extent and when we had BGC Argo floats, years when we had particularly low sea ice extent, and then we eventually decided to separately consider 2023 as the conditions observed in 2023 were quite different to other low sea ice extent years. So to start with, we wanted to broadly identify which physical and biological variables are varying with sea ice extent. So to do this, for each float, I found the average sea surface salinity in each season. And then I'm plotting it for each year against the annual sea ice extent, anomaly in that year. So get 1 point per year. And so you can see in the autumn and in the summertime, there's this clear linear trend that was statistically significant, where I'm showing the correlation coefficient squared in the panel, and it is sign preserved, which is why it's negative when there's this negative trend. And then in the winter and spring, there was no correlation. So this is telling us that in certain months, the waters are saltier when there is less sea ice. So I repeat this analysis for a large range of different biological and physical variables shown here in this table, and I've colored it to try and make it easier to interpret. So essentially, if the variable is increasing as sea ice extent also increases, then the correlation is positive or red, and the stronger the red, the stronger the correlation. And then the opposite is true for the blue. So a blue correlation indicates the variable is getting smaller as sea ice extent increases. And then if it's bold, it means it was statistically significant. So if I first touch on a couple of key things related to the more physical variables, what we found is when there was negative sea ice extent, that net heat fluxes indicated that there was more ocean cooling in the wintertime and more ocean warming in the summer. However, when we look at sea surface temperature, we'd expect waters to get cooler in winter based on these fluxes. However, what we found is that waters actually got warmer in the wintertime and spring. And so I'll touch on that a little bit more in later slides. When we look at the mixing layer depth, we found that mixing layers were deeper when there was less ice in the autumn and summer. And this is consistent with expectations because ice acts as a barrier for energy exchange between the atmosphere and the ocean. And so if there's less ice, it should make it easier for the atmosphere to mix the ocean. And so we should get deeper mixing layers. The final thing I'll touch on here is the fact that the stratification was weaker when there was less ice. And this was typically not due to changes in the thermal stratification, so the vertical gradients in temperature, but was instead due to a weakening trend in the salinity gradients in temperature. And so what we found is that the quantities that were in the mixing layer or averaged over that mixing layer had a much stronger stronger correlation to changes in sea ice extent as compared to variables that were averaged over the mixed layer or the thermocline, or above the thermocline, sorry. So this tells us it's really this thinner upper layer that is more dominated by sea ice extent. And when you get to deeper waters, other physical dynamics are more likely dominating the observations. So next, thinking of the BGC Argo quantities. So ice is highly reflective. And so if there's less sea ice, more surface part is able to penetrate into the ocean. And so this is consistent with the trend here that we found where there was more light availability over the mixing layer when there was less ice in the spring and summer. We also found that there was more nitrate in the summertime, which could indicate increased ion stress. So as I said before, we can't easily estimate ion availability as it's very challenging to observe or measure. However, if there is a change in nitrate, this could be because the more important nutrient for productivity, that is iron, was not available. We also can predict changes in quantities like dissolved oxygen and pH based on the temperature variability because their changes are related to their relative solubility in seawater for different temperatures. And what we found was that the trends were more consistent with changes in temperature for dissolved oxygen than pH and were stronger trends. And so we believe this relates back to the relative number of sensors where there was 100 less pH sensors than dissolved oxygen. So I think the changes in these more soluble variables tell us that the number of sensors that we have is of critical importance to understanding how the Southern Ocean is changing. And then last but not least, we found that there was less summer biomass and chlorophyll fluorescence in the when there was less ice, which is a key result we found. And then if you calculate the carbon to chlorophyll ratio, this tells us how much light harvesting pigments phytoplankton have, and that we found a significant result of there being less pigments in the spring and summer, which is consistent with the fact that there was more light. And so even though there were fewer BGC floats, we still found some robust and statistically significant trends. So in addition to looking at changes in the average quantity like we did in those previous plots, it's also useful to look at the distribution of data. And so we can do this by looking at box plots, where the dot here shows the median and the thick bar indicates the interquartile range, so the 25th and 75th percentile of the data. And instead of looking at all years on a linear slope or trying to fit in linear slope, instead we're picking out years that had particularly high or low sea ice and then comparing it to 2023 and the distribution of all of our floats. And so here I'm showing in the top net heat flux, then sea surface temperature, and then sea surface salinity down the bottom. And I'm showing the distribution of data in the wintertime in blue and in the summertime in red. So hopefully that all made sense. But basically what we can see from this is that even though in the table it indicated that sea surface salinities were getting warmer in the wintertime, by looking at the distribution, we can really see that if there is any change, it's really quite minimal, with temperatures on average over the circumpolar being really close to the minimum temperature. But in the summertime, there's a clear trend of temperatures getting cooler as there's less ice. And then for salinity, there's a more coherent trend of waters getting saltier as there is less ice. However, we can have a better understanding by splitting up our floats by different subregions. So we split it up by the latitude by looking at floats south of 65 and north of 65 south, where 65 south is this dashed line. And then we also split it by longitude by looking at the Pacific, Atlantic, and Indian basins. So for example, the benefits of looking at it by latitude, if we focus on sea surface temperature variability. So if we look at the more southern waters, which are more consistently covered by sea ice, even in the low sea ice extent years, less so in 2023, you can see in the winter that there's almost no variability. Almost all floats are observing temperatures that are at the minimum temperature possible in the ocean. But if we focus on the more northern waters, there's a completely different trend of waters getting warmer as there's less ice. And so that's why we observed a very small trend of waters getting warmer circumpolar, but there's a lot of value in splitting it up by latitude. And similarly, there's quite a lot of variability when we look at the different basins. So for example, if we look at the top panels for net heat flux, in the Atlantic, it observed very small net heat fluxes in the summertime in 2023. And in the Indian, it was completely different, with all of the net heat fluxes almost being larger than high sea ice extent years. If we look at sea surface temperature, we see that there's clear trend of waters getting cooler in the summer when there's less ice for the circumpolar plots is consistent to what we see in the Atlantic. It's more similar in the Indian to the circumpolar case, but then if you look at the Pacific, the trends are quite different, with low sea ice extent years having the warmest temperatures, so quite different to what the trend in the circumpolar. And then similarly in the sea surface salinity, the Indian and the circumpolar are similar. Atlantic is more similar to the circumpolar, but the Pacific is quite different. And so we observed the Pacific being different to the other basins in quite a number of different variables. And we believe this is related to the fact that the southern ACC front, as well as some other fronts, are extending into our defined marginal ice zones in the Pacific region. But by comparison, the Atlantic and Indian basin are more consistently or south of the southern ACC front, or the southern boundary of the ACC. And so I think in the Pacific, the sea ice extent isn't the dominating control, but in the Atlantic and Indian, you're more likely to see a strong difference due to a change in sea ice extent. Okay, so this is the last bit of results. And here I'm looking at changes in the timing of the bloom as well as the total biomass due to changes in sea ice extent. And unfortunately, because of the fewer BGC Argo floats, this is less robust to changes in the physical environment, particularly in 2023, because of the limited number of floats that have uploaded their data so far. So only have about 6 apexes. But showing here, there's still some coherent trends in the onset, which is when biomass is at its minimum, the climax, which is maximum accumulation rate. The apex is when biomass is at its maximum. And then I've defined here the productive period as being how many days between the onset and apex there was. And then finally, the time integrated biomass in the bottom panel, where I've integrated the carbon biomass from the onset to the apex. So in general, the trends in the 65 South region were a bit easier to interpret, so I'll just focus on those trends today. But what we found was that low sea ice extent years had shorter productive periods as compared to high sea ice extent years, as you can see with the red arrows here, with low sea ice extent years having earlier apexes and therefore shorter productive periods. And then this also resulted in the time integrated biomass being smaller. However, 2023 deviated from these low sea ice extent years, and looks like something quite different is occurring in 2023, where the apex was around a month or more later in the year, and then because of that, the productive period was also longer. However, as the tables indicated to us, there is less summer biomass when there is less ice across all years that we have records. And so even though there is a longer productive period, because that biomass is overall slightly less, what we found is that the time integrated biomass was frequently less in 2023 than in high sea ice extent years, as you can see by the difference in the 25th percentile. So even though the median hasn't changed as much, still the 50% of floats were observing biomass, time integrated biomass that was smaller than a high sea ice extent year. So we tried to put this all together into a schematic to describe what will happen in a low sea ice extent year. And so what we identified was things like a reduction in sea ice extent, an increase in sea surface temperature, increase in sea surface salinity, a decrease in the stratification, and a deepening trend in the mixing depth. And then we contrasted this with the recent IPCC report to see which of these trends that we found in our observations are consistent and which are inconsistent. And so the trends in the temperature, the sea ice, the mixing depth were generally consistent with the IPCC report. However, our observed trend of waters getting saltier in the surface layer is not consistent with the IPCC report. And similarly, our observed trends in stratification are not consistent. So that's quite interesting, and we'll be looking into that more in future. But to conclude, we found large regional variability in how upper ocean physics and phytoplankton variability respond to changes in sea ice extent. Overall, the more northern waters, as well as the Indian Basin, were most sensitive to changes in sea ice extent anomalies. And we found the Pacific Basin to be the least sensitive. And we attribute this due to the fact that a number of ACC fronts penetrate into that marginal ice zone in the Pacific Basin, and they don't so much in the other regions. We also found observed physical variability was generally consistent with the IPCC report. However, the sea surface salinity and the stratification was inconsistent. So interesting things to look into more detail. So I motivated this work by saying that I wanted to look at hydrotrophic waters, as well as things like phytoplankton community composition and other details of the biology. However, BGC Argo floats are quite limited in what you can understand with these other higher order variabilities or more details on the biology. And so if we want to look at the more details on the biology, we unfortunately need to focus on a region where there is more comprehensive observations. And so we're hoping to do that in future work. And related to observations and sampling, we note that there was a paper in 2014 that identified that we need about 200 BGC Argo floats in the Southern Ocean to adequately monitor change. And by looking at physical variability, where we have over 200 floats, we can see clear trends that is relatively robust. But unfortunately, we only have about 40 BGC Argo floats in recent years. And so it was a lot harder to identify statistically significant trends, and is a real shortcoming of this analysis. And so I believe it's critical that we continue to maintain the BGC Argo program. Ideally, we want to have more floats if we want to understand how the Southern Ocean is changing in the future. And I also advocate for expanding the sensors because the few sensors we have that have additional sensors like irradiance or dissolved oxygen or pH really add to our understanding of the Southern Ocean. Okay, thank you for your time. Hopefully there's some opportunity for questions.
That's awesome. Thanks so much, Tamara, for a great talk. Yeah, well done wrapping your head around all of those incredibly interactive factors. So it looks like David has a question. Do you want to go ahead? Sorry, we can't hear you, or I can't hear you.
It should work now.
Yeah.
Okay, sorry. Thank you, Tamara. Very nice talk and very nice work. I just have a quick question on the first part of the talk. Maybe I missed something. I don't remember if you look at all this problem of the blooms, looking at the surface chlorophyll or the integrated content, and whether you tried as well to derive some growth rates, because this coupling decoupling theory is largely based on the growth rates, I think. So that's the question I had. And another one, sorry, is about this mixed layer average irradiance. I was a bit surprised by the range of values you had, so I would be interested to understand how you did calculate this. Thank you.
Sure, thanks, David. Yeah, so I mostly focused on the optical backscatter because, at least in the Southern Ocean, if you have it available, it can be a more reliable estimate of the biomass. As you know, the chlorophyll fluorescence sensors are highly sensitive to the photophysiology. So essentially, the more light harvesting pigments they have within a cell, the chlorophyll fluorescence sensor will observe a change, even though there's no actual change in the phytoplankton biomass or like the carbon content of the cell. That's my very simple description for everyone. And so for that reason, I focused on the optical backscatter because, yeah, we expect there to be a change in the photophysiology and therefore the chlorophyll fluorescence around the wintertime when the light is so minimal. And so we find a change in chlorophyll that was not due to biomass changes. However, there's some interesting work by that Pete's been involved with that has looked at differences between chlorophyll and biomass. So I'm happy to point you to those if you're more interested in the chlorophyll variability. And for your second question, some sensors in the Southern Ocean do have irradiance sensors, but they do a sample every 5 days or 12 days, 10 days on average. And so it's impossible to get an estimate of the daily PAR from just individual samples like that. And so instead what I did is I used the irradiance sensors or chlorophyll fluorescence, if we don't have irradiance, to estimate the diffuse attenuation of light. So essentially how rapidly light gets absorbed as it penetrates the water column. And so that was the one part. And then to get the surface PAR estimates, I had to rely upon the ERA 5 data. So I basically took the shortwave radiation, I applied a bias correction based on Mark Mallett's work, and then I used the diffuse attenuation that I estimated from the Argo floats to get a penetrating irradiance. And so, there are limitations in that work, but that's basically the best we could do.
Thank you.
Great stuff. Anyone else have any questions? Please feel free to unmute and ask it.
Hi, Tamara. Oh, I have a question.
Yeah, Zana, you go, and then Rob afterwards.
Yeah,
Thank
sorry,
you.
I just saw Rob's hand go up. I just, that was a great talk. Thank you very much, Tamara. I was curious about why the mixed layer depth is so often not aligned with the mixing depth. Like, why don't they align so well in the Southern Ocean? And if that's particular to this region or if it's kind of more widespread than we realize elsewhere.
Yeah, so I guess it's not always important to distinguish a mixed layer from a mixing layer. Like it depends on what your question is, like your research question. But yeah, I think the typical definitions for a mixed layer of that, looking for a density difference of 0.03 is like one of the more common ones. It kind of tells us where, like some definitions call it the remnant mixed layer. So it's less variable in time. It tells us where mixing has occurred recently, and the active mixing depth you can identify by with relative confidence by using a much smaller density gradient of 0.005, and so this gives you a lot more confidence where the mixing rate, if you were to measure it, is likely to be 4 orders or 2 orders of magnitude larger, and so in the Southern Ocean, that Magdalena Carranza paper was saying that these storms come through about every week. And so when the storms come through, the mixing and the mixed layer depth are the same. But then as time goes on, the active mixing depth will rapidly show, but it takes a lot longer for the density to re-stratify. And that's why there's such a divergence of these two depths. And the reason it's important for things like phytoplankton is that phytoplankton are able to, they can re-stratify, and the fluorescence quantity, for example, will re-stratify at a much faster rate. So that's for certain quantities, it's important to distinguish between the two. Does that answer your question?
Yeah, no, that's really helpful. Thank you.
Great, thanks, Anna. Rob, do you want to go ahead?
Thank you. Thank you very much, Kieran. Thank you, Tamara. Couple of questions, please. The first one is, how do the floats measure sea surface temperature in an ice-covered area?
Yes, good question. So that is something I didn't talk about. Yeah, so technically I'm not really measuring sea surface temperature. What I'm showing there is the most surface, like the shallowest temperature measurement. And so BGC Argo or Argo floats have ice avoidance software. So if they hit that, minus 1.8 temperature or around that, they immediately will start going down instead of continuing to the surface, because if it's getting that cold, it's more than likely going to have ice there present. And so because of that, yes, the sea surface temperature might be different at the very surface or just below the ice, and the float isn't going to get close to there. So I am assuming that minimum, close to minimum temperature is representative of what's happening just below the ice.
Great. Thanks very much, Tamara. And excuse me, the second question may be a silly question, but to what extent are phytoplankton blooms seeded by ice algae melting out of the ice come late spring and summer? I'm sorry if that's a silly question.
No, it's not a silly question. Yeah, it's not so much my forte. I've seen some contrasting findings in the literature. I know it's an active field of research with some people here in ACEAS also quite interested in that problem. And we think it's probably important to understand, particularly when biomass is at its minimum and concentrations are very low in the winter and spring. But my understanding of the problem is that we don't really know how important sea ice algae is because we have limited observations. And so some of the recent field campaigns near Antarctica will hopefully tell us more about it. But yeah, that we don't know whether or not these ACEAS algae are like just drifting down to the deep, or whether or not they are an important contribution to the bloom. But that's my understanding. Maybe someone can correct me.
Thanks, Tamara.
Yeah, thank you, Tamara.
Joao, do you want to go?
Hey, thanks for the talk. I'm a question about, have you calculated the vertical distribution of the buoyancy flux, say, which represents the stratification of the seawater? Since comparing the vertical distribution of buoyancy flux and the phytoplankton concentration can better study the impact of temperature and salinity changes on phytoplankton vertical distributions. You know, sometimes rejustification occurs in the mixed layer, which can affect the vertical distribution of the phytoplankton concentration.
So sorry, you're asking if I've looked at the depth through depth variability in stratification, and particularly the role of temperature and salinity, was it?
Only consider analysis the temperature and salinity. We don't know the stratification. So if you calculate the buoyancy flux, which denotes the stratification of the up there waters. So I think maybe the bug can use the data can use calculate the stratification in the upper ocean. So I think this variable may be suitable to study why the vertical structure of the phytoplankton distributions.
Okay, thank you. Yeah, so I have because I'm working with a large number of floats, I haven't so much been looking at the through depth data, and I've more tried to identify things like the depth of the mixed layer, or the strength of the stratification, and compare them across floats. So I found that in the more southern waters, there's more consistent trends between when the stratification is changing and what's happening with the phytoplankton, I think, and less so as you get more north. Yeah, so I think I haven't estimated the buoyancy flux per se, but I think I've looked at quite a bit at the stratification, which I think is similar. And yeah, the expectation for the Southern Ocean is that the stratification is going to increase, and because of that, there's going to be less nutrients available in the surface waters. Maybe less so for the polar, but like that's how the oceans are predicted to change. And so yeah, it was somewhat surprising that the stratification was we found was decreasing as there was less ice. And we're not really sure. I guess I haven't thought enough about how that could impact the phytoplankton, but I'll have to think about it some more. Thank you.
Yes.
Thanks, Tamara. You're dealing well with lots of difficult questions. So, does anyone else have any? Or I just have a little comment. Or I really liked your schematic, kind of aligning how your results agree with the IPCC kind of projections. And you mentioned future work. Are you thinking about ways that you can use the kinds of hypothesis testing that you're using to maybe plug into some of the IPCC models to see kind of the divergence of phytoplankton across the region?
Yeah, that'd probably be a good idea. Yeah, I guess I'm not as familiar with the climate change literature, so I've been trying to familiarize myself with that. I guess if you have studies, if anyone has studies that they recommend me to look at, I'd be very interested. Because I guess the IPCC report seems to be like a composite of lots of analyses, right? And then they attach a confidence level based on how if all papers it's pointing to one thing, then that's high confidence, and if they're all different, that's low confidence, for example. And so I guess that's why I thought it was good to look at the IPCC report, because otherwise a lot of studies are more like regional based. But if anyone has any papers or things that they recommend me to look at, I would be very much like to hear your advice. Thank you.
Great, thanks. Any other questions or comments for Tamara, or helpful paper suggestions? Or I'm sure you'll be able to find her email pretty easily. Okay, yeah, if there's no more, then we might wrap up. It's nice to have a couple of minutes before if anyone else has another meeting at 2 o'clock. So maybe just can join me again in thanking Tamara for a great talk. And yeah, if you want to reach her with any follow-up questions, I'm sure it's the standard UTAS email, so your name with a dot in the middle. Great, awesome. Thanks so much, Tamara.
Great, thank you, Kieran, and everyone for your time.
Cheers, and good luck with the house settling and everything. Okay, thanks everyone. See you next time.